Hi, in this video we'll continue our discussion about chromosomes, talking about their structure, the three essential parts of a chromosome. We'll talk about chromatin, which is the DNA chromosomes and the bound protein. We'll talk a lot about histone proteins, which are the primary proteins which package DNA inside the cell. And we'll talk specifically about the tails of the histone proteins, the very ends of them, which play a very important role in regulating gene expression. In this video and the next several, we'll be talking primarily about events that are happening during interphase. That is the part of the cell cycle that is not directly involved with mitosis, which we'll be speaking about at a later time. During the cell cycle, the chromosomes exist in different configurations. During interphase, which is most of the time in which the cell is doing work, the chromosomes are decondensed, they're uncondensed, they're spread out as far as they go. It's during mitosis when the chromosomes will compact incredibly and will form the structures that you can see here. There are three essential components to any chromosome: the telomere, replications of origin, and the centromere. We've already met the centromere when we saw pictures of karyotype and the way that the chromosomes were aligned. The centromeres are the center part of the chromosome, and it'll be to these features which the mitotic spindle will attach and will help pull and separate the two chromosomes from each other during cell division. The telomere is the ends of the chromosome, and they have a specific structure that helps protect the linear ends of the chromosome from degradation. Additionally, they play an important role in regulating the lifespan of normal cells. The third important part is the replication of origin. Each chromosome has many replications of origin. It's essential that there is at least one replication of origin on either side of the centromere. As the name suggests, this is the place where DNA begins replicating itself. We'll discuss each of these pieces in further slides. On this slide, you can see a detailed image of an interphase nucleus where the chromosomes are at their most unwound. Even in this state, you can see there is some structure with the way that the chromosomes are organized in the nucleus. Different colors indicate patches where a particular chromosome dominates. We don't yet understand the importance of the relationships of the locations of the different chromosomes, and whether it's unchanging, varies from person to person at time to time, or it's a fundamental property of the organization of the chromosomes. But it does seem clear that the organization is regulated in some manner, so there must be a reason for it. At the bottom here, you can see the electron micrograph of a nucleus. The borders of the nucleus are at this barrier right here, and within the nucleus there's an area of dark color, which is the nucleolus. The nucleolus is an area of intense activity. It's here where the ribosomal RNA and the RNA for ribosomal proteins is synthesized. Ribosomes are some of the most abundant proteins, and so they need to be made in great quantity throughout the cell's activity. On this slide, you can see various levels of organization that the chromosome undergoes as it's packaged from its free form, which extends a few meters, to its most compact form, which is about 10 microns in the longest dimension. I have that at about a 100,000-fold ratio, but the point is that it's incredibly condensed, and the way that the cell achieves this is quite remarkable. We'll focus mostly on the top levels of organization, and in particular we'll be talking about this one, the so-called bead on a string level. In this view, you can see the red DNA is wrapped around yellow proteins. Those yellow proteins are the histone proteins that we were talking about. And this picture is reasonably to scale. The DNA winds around histone proteins about twice and is separated from other histones often by about the same characteristic length as the histone is wide. A nucleosome refers to one set of histone proteins where the DNA wraps around approximately 1.8 times. This represents one of the beads on the bead on the string structure. The protein core is made up of about 4 different histone proteins, colored in green, blue, red, and orange. And there's 2 copies of each, so there's actually 8 protein subunits on the inside of a protein core. A 5th histone protein, H1, binds to the outside of the nucleosome, but it is not shown in this picture. Each of the 8 protein subunits has this tail region, which can wrap around and bind the DNA very tightly. It does so by possessing a lot of positive charges, a lot of lysines and arginines on the cysteine tail, that will interact with the negatively charged DNA very strongly. The other 8 tails of the histone proteins, the other 7, are not shown in this diagram. Here we have another picture of the banding patterns on chromosomes. And we can now say that the light regions, or the ones that appear white in the picture, is referred to as Euchromatin. Euchromatin is less compact, it's more accessible to proteins, and in this case to fluorescent dyes which are added to the preparation. The dyes are able, because the chromosome is less tightly wound, the dye can get in there and it will bind to the DNA, intercalating, and so that it is able to be concentrated there and it can fluoresce very brightly. The dark regions are called heterochromatin. These are wound more tightly, there are fewer genes, and the genes that are there are more often than not inactive. Switching between these 2 regions can be achieved by modifying the positive charges that are on the histone tails so that they will bind DNA less tightly and the DNA will be less tightly wound. To switch between the two forms of chromatin, highly condensed or more open, one has to remodel the chromatin. This is achieved by the input of energy, the hydrolysis of ATP, and rather than removing histones, they are slid along the DNA to have greater spacing between each other. This is the configuration that the chromatin is likely to be in when it contains an active region with genes that are being transcribed. I think it's pretty remarkable that when the polymerase will read the DNA and transcribe it into RNA, the histones are present, but they're just even looser, and that the polymerases will navigate around the histones. Now on this slide, we see a schematic of a histone protein in yellow, drawn extremely not to scale. Here's the carboxy terminus, here's the amino terminus. This is a blow-up of the histone tail region. As you can see, there are many lysine residues, 6 lysine residues that's indicated by the letter K, which is the 1-letter code for the amino acids. Now, lysines are positively charged, normally. When you have many unmodified lysines, you have all these positive charges interacting with the negatively charged DNA. And as we've said many times already, opposite charges attract, so it's a strong interaction. This makes it difficult for the polymerase and other transcription factors to access the DNA. When the lysines are acetylated, that neutralizes the positive charge into a neutral state, and now a neutral histone tail will interact less strongly with the negatively charged DNA. And in this case, genes in this region of the chromosome are turned on. There are many intricate details about the modifications of the various residues in different locations of the histone tail, and this is referred to as the histone code, which is still being elucidated. But for our purposes, we'll stick to the very basic point that if histones are acetylated, in particular the lysine residues are acetylated, that the genes are turned on, whereas if they are in their native state, their naked state without acetylation, then there'll be a great deal of positive charge and the genes are turned off. In this slide, we see a picture of the structure of lysine with the positively charged amino group at the end of the side chain connected to the alpha carbon. In the deacetylated state, you have the positively charged amino group and the negatively charged phosphate group of the DNA. If the lysine is acetylated, here's the amino group, here is an acetyl group. This neutralizes the positive charge. Now there's a negative charge, but no positive charge here, and the interaction is much weaker. On this slide, we see a schematic of a Drosophila chromosome. We see the centromere, the two telomeres, one on each end, and we see regions of heterochromatin alternating with euchromatin. Euchromatin, this is the light band on the chromosomal karyotypes. These are the dark regions, the heterochromatins. And this is where the active genes are, and this is where if genes are in this region, they are inactive. These regions are maintained and regulated by chromosomal elements referred to as barriers, which can keep the heterochromatin structure from spreading out into the euchromatin region. In some cases, you have DNA anomalies where the chromosome is inverted, and now there's no barrier between this gene and the heterochromatin. And as this picture suggests, there are phenotypic consequences that result from the altered locations of the gene. And this manifests itself in the pigment of the eye color. And while it's slightly confusing that if the white gene is silenced, that white would appear on the eye, but nevertheless that is the case. There are many other instances like this, and almost any gene located near a centromere or a telomere is almost always less active than genes that are some distance from either of those two structures. Okay, thanks for listening.